Hi, everybody. I'm Tatyana, CEO and co-founder of ImYoo, and today I'm going to walk you through something really exciting, your ImYoo Immune Report. When you participate in the Patient Scientist Network, you're not just contributing research, you're getting something valuable back, a detailed look at your immune system. Today, I'll show you what that report looks like and what those colorful graphs actually mean. So let's dive in. We'll start in the lab. Let's quickly talk about the different steps we do in order to get your white blood cells into a data. Step one, we pull out the white blood cells from your blood sample when it arrives to our lab. These are your immune cells. They're the ones that are doing all the immune work in protecting you, making sure you're healing up. Step two, we take out about 3 to 5,000 of those cells and run them through a machine that measures which genes each cell has turned on and off. This is what we call single cell RNA sequencing. Step three, from that data, we figure out what type of cells they are, T cells, B cells, monocytes, natural killer cells, and we compare how your cells are different from other participants and to yourself across time. Pretty cool, right? Okay, now for the exciting part. Let's look at your report and what all these colorful graphs mean. So once you've got your email and consented to everything, you should get your participant report. We version this report in case we change any of the analysis and update anything. We can send you a new report. All right, let's take a look at subject 3. So the first thing you will see here is this plot, which is called a sunburst plot. What it shows you is a distribution of different cells within this particular sample, or an average of samples if you took more than one. So what you see here, we start at the inner circle. These are your more base T cells. We call them T cells, B cells, natural killer cells. And it shows you the different distribution. For example, this person in their sample, 81% of the white blood cells that we detected were T cells. So as you go further out into what we call the sun or the circle, you start to see the subtypes of different T cells. There's a lot of them. So you see CD4 T cells and CD8 T cells, and you can see in terms of percentage how much of them make up the total T cells. So this is interesting to see. Standing alone, it doesn't really mean much, but it's interesting to look at your distribution of different cell types. So going further, you're going to see all these dots overlaid on what looks like a map. It is a map. We're recording a lot of molecules, 3 to 5,000 cells by 10,000 or more genes. In order to actually visualize that, we need to project it in 2 dimensions. So what you see here, the orange dots are your cells, and the gray map-looking thing is everybody else. Nothing actionable here, more of an abstract, fun visualization, just to show how big the data is. So going further, here is where you see your cell type abundances. So this is how much of a cell type you have compared to other people in the ImYoo atlas. So this is compared to everyone who is in the ImYoo database. So if you had multiple samples, you'll see red or blue dots based on whatever state you were recording. So this person is a red dot here, and you can see here, hey, this person has more T cells compared to average in the ImYoo atlas. And then as you go down further, we break the cell types. As I said, there are many different types of T cells, and this breaks it down further. So CD4 T cells, CD8 T cells, different types of B cells. You can kind of geek out and explore and dive into that. And the reason we're able to see so much precision in terms of these cell types is because we record so many molecules, and that allows us to see much deeper into the different cell types. So as I said, we're not just measuring what kind of cells you have, but what are those cells doing? So here you're going to see in section 4 a thing called differential expression. So this is where we are comparing either you to everybody else in our atlas, or if you participated in a study where you collected multiple samples, you're going to be comparing your say healthy state to your flare state if you're in our autoimmune flare study. What's interesting here is, so you're looking, for example, this person has higher expression of this gene compared to everyone in our atlas in CD4 T cells. So the outskirts is where the interesting things are happening. Remember, this is research, so nothing here is clinically actionable or, yeah. So, and as you go further down, you can see different cell subtypes, such as CD8 T cells, gamma delta T cells, so you can geek out and look those up. And then in the last section, we measure many genes, as I said. So we took a few of them, kind of a little bit like a Snapple fact, if you know what those are. We're looking at different known inflammatory genes. or immune-mediated genes, IL-6 being one of them. So we tell you a little bit of a fun fact and show you how much of the gene gets expressed in different cell subtypes. Um, and like I said, we're doing this for several genes just so you can kind of see, what that looks like and hopefully learn a little bit about these. So just to conclude, we want to say thank you for participating in the study and creating this research data. We hope in the future it can really be used to help you personalize your health better, especially as you measure these samples against yourself and try to understand how's your immune system evolving. Um, as I said, keep in mind this is research. If you have any questions, please ask us. You're welcome to take this to your doctor and geek out with them. And yeah, let us know if any questions, thoughts, ideas. We welcome it all. Thank you again.